started. As a reminder, those test corrections, if you get any of those specific questions wrong, those are due on Friday. So you have some time, because I know some of you didn't get your test back until today. But we're going to move on with cellular respiration today. And so we talked about cellular respiration a little bit last Friday. We talked about that first step, glycolysis, right? And we're going to talk about the remaining 3 steps today. So lots of information, I understand. But before we go on, I just wanted to do a quick review of glycolysis, right? Because it's been a weekend, right? It was family weekend, and some of the family on campus, and cellular respiration was the last thing on your mind. So we did talk about glycolysis, right? So we're creating these 2 pyruvate molecules, and this is happening outside of the mitochondria in the cytosol, right? And so what I want you all to do is just take a second and do some review on your own. Look back at those notes. Think about glycolysis. What are the 3 major stages? What does each stage produce? And what is the net overall ATP and NADH yield of glycolysis? Yes, but I'm not sure. All right, I started my timer a little late, so let's go ahead and discuss what are those 3 major stages of glycolysis? Yes, energy investment is one of them. What is the second one? Cleavage. Yes, and the third one? Yes. Oh, I apologize. I did not catch that mute on that one. That is my fault. And now I have no idea where it went. All right. Well, that was a fun wake up. All right, maybe we needed that on a Monday, huh? So, yes, we have energy investments, we have cleavage, and we have energy liberation. All right? And those major molecules produced in each step, there's not much for this energy investment step that I want you to know. But we do create this fructose 1,6-biphosphate. Right? It was not bolded because I don't expect you to recall that. But I do expect you to know that we are using 2 ATP in that energy investment, hence the investment part of that phase. Right? In cleavage, we're breaking 6,6,6,1,6-biphosphate, right? We're cleaving it apart, hence the name, into molecules of G3P, glyceraldehyde 3-phosphate. Yes. So glyceraldehyde 3-phosphate. Right? So now we have 2 glyceraldehyde 3-phosphates. And then in this last step here, right, those glyceraldehyde 3-phosphates, they turn into pyruvate. Right? And in the meantime, we are producing 2 NADH and 4 ATP. Right? Now, even though we're producing 4 ATP here, we're using up 2 ATP in that first step. So the net yield is 2 ATP and 2 NADH. Yes? Intermediate molecule. I'd have to look it up to see what it's like used for other than cellular respiration. I only know it as an intermediate in cellular respiration. I'm sure there's probably other ways that the cell might utilize it as well. Any other questions about glycolysis? All right. So I did mention last week that we have to maintain homeostasis within our body. We often do this by feedback inhibition, right, where the products inhibit the reaction from proceeding any further. And we see that in glycolysis as well. So in the third step, I know this is kind of a very small image here, but in the third step, we're in that energy investment phase, right? This phosphofructokinase catalyzes that third step in glycolysis. And we have feedback inhibition here, right? So we're producing ATP in the last step, and when we have too much ATP, it's going to inhibit the function of this enzyme, and thus stop glycolysis from occurring, at least temporarily, because we have ATP, we don't have to invest more energy into making more when we don't need it, right? So feedback inhibition is occurring in glycolysis. Okay. One more thing about glycolysis before we move on is that just kind of a fun fact, right? So cancer cells, they typically have high levels of glycolysis, right? This is known as the Warburg effect, right? So these cancer cells, they prefer doing glycolysis and only glycolysis. They won't go on to the rest of cellular respiration, such as oxidative phosphorylation, which we'll get to in a bit here. But this can be useful in diagnosing cancer, right? So in this image here, right, this individual has ingested some glucose molecules with a fluorescent tag attached to it. Right? It's not harmful to us, but it allows the imagery to pick up where those glucose molecules are concentrated. And we see in this individual, they are highly concentrated, kind of around the spine here, right? I'm not a radiologist, I don't know how to correctly read this image, but big picture here is where they are concentrated is where we find tumors, right? Further kind of going into this effect of tumors preferentially using glycolysis and not going on to the rest of the cellular respiration cycle. So, a little bit of a fun fact. I won't be testing you on this, but I thought it was interesting to talk about. All right, but let's get into some new material. So at this point, we've gone through glycolysis. We have 2 molecules of pyruvate. We now have to process this pyruvate. Right? So the second step, we call it the breakdown of pyruvate. All right? It's step 2, and in step 2, pyruvate is transported into the mitochondrial matrix, right, into the middle of our mitochondrial cell. So we're no longer outside of that organelle. Excuse me, I said cell. It's organelle. No longer outside of the organelle. We're now inside of the organelle, and that's where the rest of our reactions will take place. Okay. Once it is inside of those mitochondria, a couple of things are going to happen. Right? We're going to break down pyruvate via the enzyme pyruvate dehydrogenase. Okay? So we're breaking down our initial pyruvate to make this intermediate molecule. Okay? In the process of breaking down pyruvate, we are producing CO2 as a byproduct, and we are oxidizing NADH. right? So we're creating some NADH. All right, we can see that here we're producing CO2 and NADH. All right, now I don't expect you to know the names of the intermediates. It's not even labeled on the graph. I don't deem that as big picture importance for Bio 101 here. But I want you to know that it's broken down, right? We're producing one NADH for each pyruvate molecule. And remember, glycolysis produced 2 molecules of pyruvate. And what happens in that very last step is that what's left of the pyruvate molecule is an acetyl group, and that acetyl group is attached to a coenzyme to make acetyl-CoA. Right? Coenzyme is a molecule that basically helps an enzyme out. Right? It's a very simplified way of putting it. Right? So that's the breakdown of pyruvate. Pretty simple, pretty not too much going on here, right? Big picture, we are getting one NADH, and we are prepping this molecule for our next step. All right, our next step is the citric acid cycle, also known as the Krebs cycle. Right? I like to call it the citric acid cycle. Right? So this is a cycle, right? Glycolysis is very linear. Breakdown of pyruvate is very linear. So the Krebs cycle, it's a cycle because the molecules will get regenerated and used over and over. Okay? So in this cycle, some molecules are entering, some molecules are leaving. Okay? And it's a series of molecules that are being regenerated in each step. So the very first thing that's happening, right, this might be as much detail as I want you to know in terms of the different molecules, right, is that our acetyl-CoA, that's the product after the breakdown of pyruvate, right, the acetyl is removed and this CoA leaves the reaction, right? That acetyl is going to combine with this molecule called oxalacetate. And once it combines with oxalacetate, it becomes citrate. Hence the name citric acid cycle. All right? So these 2 molecules are pretty important. I want you to remember these. All right? So acetoacetate, I always stumble over all of these words, and today I'm just having a jumbled mouth or something. Oxalacetate is combined with the acetyl group to form the citrate. This citrate is going to go on to the rest of the cycle and go through a series of reactions. Right? Throughout these reactions, we are producing CO2 as a byproduct. We're producing a molecule of ATP, as well as NADH, and this molecule called FADH2. Now, we have not talked about FADH2, but think of it as kind of like a sibling to NADH. It does the same thing as NADH. It's just the molecular formula is a little different, right? It still carries electrons and hydrogen atoms like NADH does. All right, so I'm not going to go into too much detail about that molecule. You can think of it working exactly the same way as NADH. All right, after the cycle has ended, right, the last step actually regenerates this oxalacetate, and then the cycle can begin again. All right, so let's review it again with a different image here. All right, so we are inside the mitochondrial matrix here. We have this cycle that's happening. I really like this image because this image has all the information on it that I would expect you to know. All right, notice the only 2 molecules that are labeled are the oxalacetate and the citrate. All right, we have this acetyl group coming in. The coenzyme A is leaving, and the acetyl group joins with oxalacetate to form citrate. All right. We then have all these other steps that are happening. All right. What I want you to notice about the steps is not the specifics, but what is being used and what is being produced. All right. So citrate, we're going on to these other steps. We're producing molecules of NADH. We're producing CO2. Right? We're producing a molecule of ATP and a molecule of FADH2. Right? That last step, we're also producing an NADH, and that step is what regenerates this oxalacetate. Right, we're going to zoom in on that regeneration of oxaloacetate, the very last step in that cycle. Right, so oxaloacetate, it's accepting that acetyl group, and then it changes form throughout that cycle. Right, but in order for this cycle to continue, we have to get the original oxaloacetate back, and that's what this last step is doing. Right, we're taking malate, which was formed the step before oxaloacetate. Right, we are oxidizing NADH, right, and in the process, those hydrogen ions are coming off of malate, going on to NAD plus to form NADH, and that leaves behind our oxaloacetate, right, to be used again in the exact same manner. Do we have any questions so far? Yes. Yes. No, excuse me. Oil rig. Oxidation is losing. I know, it messes me up too. Yes. The COA. So the COA leaves glucose. Yep. So the coenzyme A is added at the very last step of the breaking down of pyruvate, but then it's immediately released once it goes into the Krebs cycle. Right. Any other questions? All right, let's think big picture again. Right. So I would want you to know what the products are for the citric acid cycle. Right. And I want you to know what it would be for one turn of that citric acid cycle, but also one molecule of glucose. Right, now remember, glucose is split into pyruvate and glycolysis. Right, so we have two pyruvate molecules that then turn into two acetyl-CoA molecules, and each of these acetyl-CoA molecules go through the Krebs cycle. Right? So for one molecule of glucose, right, it's kind of like two turns of the cycle, we would get 4 CO2 molecules, 2 ATP molecules, 6 NADHs, and 2 FADH. Right? That's just the citric acid cycle, nothing previous. Right? Does that make sense? It's hard to read your faces today. It must be a very tiring Monday. All right, so that's most of cellular respiration. All right, so at this point, glycolysis, breakdown of pyruvate, citric acid cycle, the total of molecules produced: 6 CO2, 4 ATP, 10 NADHs, and 2 FADH. Right. Now notice that's not a lot of ATP. Right? And the purpose of cellular respiration is to make ATP. Right? We're kind of generating our molecules up until this point so that we can create ATP. Now we have a little bit produced, but notice this NADH, we have 10 molecules of NADH. Okay? And this is what becomes important in our next step. But I want us to take a step back and kind of digest what we've already learned. Okay? So I thought a good way to do that would be to do some research of what happens when we have a dysfunctional Krebs cycle. Right? So dysfunctional enzymes or intermediates in the Krebs cycle can lead to a group of rare genetic disorders known as mitochondrial diseases. Right? These disorders are characterized by impaired mitochondrial function, which can result in a wide range of clinical manifestations or symptoms. Right? So I want you to find a mitochondrial disease. I want you to tell me what causes this disease, right? How is the Krebs cycle impacted? And what are the symptoms of that disease, right? And in the meantime, it would be a great time to ask me any questions about the citric acid cycle. I really hope the police siren sound is off on this one. It is. Good. All right. So, what are some of those mitochondrial diseases that we found? Say that again? Leigh syndrome. What happens in Leigh syndrome? Okay. Yeah, so we're affecting the Krebs cycle, making it slower, and so symptoms might be aging milestones a little, hitting those milestones slower. Yeah. What other disease did we find? A whole handful of them. All right. Well, maybe we just need to go on. I can't recall them off the top of my head. I was relying on you all to recall them for me. So I know Leigh syndrome is one. I think there's maybe another 4 or 5 out there that are common in terms of rare diseases. But they all affect the Krebs cycle in some way, shape, or form, and we can have various symptoms, right? All right. So this is our last step, right? And this is a step we often kind of get hung up on. And so I'm going to try to break it down simply, but please let me know if you have questions. So this is the last step. We call it oxidative phosphorylation. All right? This is where we are utilizing the NADH and the FADH2s that were produced in the previous steps in order to make ATP. Right? So this is the stage of cellular respiration where we get that big ATP payoff. Right? So we're removing getting electrons from NADH and FADH2. Right? This step typically requires oxygen. Right? Oxygen is a final electron acceptor here. And when it accepts electrons, you get water as a byproduct. And it involves the electron transport chain. So when I say the electron transport chain, I am talking about a series of proteins that are embedded in this phospholipid bilayer inside of your mitochondria. Right? So we have a membrane around our mitochondria, and then inside of that initial membrane, we have another series of membranes, right? The mitochondrial membranes, and that membrane is where our electron transport chain is located, right? Where we have these proteins embedded within that bilayer. And then, so basically, big picture here, we have these electrons, we're creating a gradient, and then we are using that gradient to power this protein called ATP synthase. And it's that protein that really makes the ATP for us. All right? So let's talk about building up that gradient. Right? So again, we're in that inner mitochondrial membrane here. Right? We have our proteins embedded, and we have an abundance of NADH and FADH2. Right? They are carrying these hydrogen ions for us that we are then going to strip off of those molecules and create a hydrogen ion gradient. And so if we look at this image here, you see we have NADH interacting with one of our proteins. The hydrogen ions are being stripped off and transported across our membrane. We also have FADH, right? Those hydrogen ions are also being stripped off and transported across our membrane. Right? Now, I don't feel like it's important in Bio 101 for you to memorize all the different protein complexes and the very intricate stages of how this happens. So again, I want you to think big picture. I want you to know that we're using these proteins to strip away these hydrogen ions to build up a gradient. Where now we have more hydrogen ions in the intermembrane space than inside of our mitochondrial matrix. Right? It's this gradient, right? This gradient's often called the proton motive force, right? Hydrogen ions are often called protons, right? It's this gradient that powers our ATP synthase, right? So our ATP synthase is this very complex protein. It has a lot of different subunits to it, right? And it's this protein that can phosphorylate ADP molecules. When I say phosphorylate, I mean the addition of a phospho group, right? So if you remember ATP, we can split apart one of those phosphate groups, releasing energy, and now ATP becomes ADP, right? In this step, we're taking ADP and turning it back into ATP by adding back that phospho group, right? Now it's important to note here that even though it's a relatively small ion, it's still an ion, right? So it can't easily go through the phospholipid bilayer. So this ion has to be transported through a protein. And that protein is this ATP synthase. Right? And so as this hydrogen ion is being transported through this ATP synthase, right, that ATP synthase is capturing that energy associated with this hydrogen ion being transported through. Right? So it's capturing that energy due to the movement of our hydrogen ion. Right? That energy is captured and then put into the bonds of ATP. Right? Notice we have ADP plus a phospho group. Right? We're putting that energy into the bond here to create ATP, which is now a high energy molecule that could be used in many other cellular functions. Right? It contains that energy within its bonds. Right? So as this hydrogen ion moves through, right, this is actually a pretty dynamic protein here, and that it will rotate all these different subunits as it produces ATP. Right? Pretty cool. We can go into really some in-depth stuff here, but Yes, yes. It's the movement of this ion has energy to it. That energy is being captured and put into that covalent bond of ATP. I know, it's a lot. It's a pretty cool mechanism. So, I mentioned that this ATP synthase moves. Right? So it has these different subunits. This is a very simplified version here. You can see subunit 1, 2, and 3. ADP and a phospho group will enter into one of the subunits. It'll rotate, right? Now it has subunit 2 up top. Make ATP, rotate again, and then release the ATP. Right? So it's a very dynamic protein. All right, that was a lot. And I do really well with interactive visuals, so I thought we would watch this nice video and just a synopsis of how this happens. He does go into a little bit more detail than I would expect you to know, but again, focus on the big picture here.
Once NADH feeds electrons into the first component of complex 1, these proteins facilitate a series of redox reactions, shuttling the electrons downhill from one component to another, with each structure down the chain having a higher affinity for electrons than the last. This process does not generate ATP directly, but a byproduct of this series of electron transfers is the generation of a proton gradient across the membrane. Protons accumulate outside of the inner mitochondrial membrane, which then go to power another protein complex called ATP synthase. As you might guess from the name, this is the component that synthesizes ATP. Because the proton concentration becomes greater in the intermembrane space than inside the mitochondrial matrix, the protons will spontaneously move with the gradient to re-enter the mitochondrial matrix, and the only route available is through ATP synthase. This process is called chemiosmosis, and because the gradient is able to do work through chemiosmosis, we also call this the proton motive force. And it is these protons that power ATP synthase in phosphorylating ADP to generate ATP.
All right, so I really like that video because it gives you a nice visual and overview.
Once NAD.
All right, so it's that last step, that stage 4, that oxidative phosphorylation that creates all of that ATP, right? We're really utilizing NADH molecules here and a little bit that FADH2 molecule, but we don't produce as much FADH2 as NADH, right? Once we have all that ATP, right, we could get up to 30 to 34 ATP molecules per glucose molecule. Right? That's a lot of ATP for one glucose molecule. Right? Now I'm giving you a range here because often we don't end up making as much ATP as we possibly could because there's other stuff going on in the mitochondria, right? So NADH, while most of it's used to make ATP, it can be used in other pathways in mitochondria. Right? So these anabolic pathways building up different molecules. Right? It could be used, once it makes that gradient of protons, right? That gradient can be used for other purposes. Right? So not all of the NADH and not all of that proton gradient is being used solely for ATP, but a lot of it is. Yes? Great question. I would guess, and I'm going to make a note so I can put this up here, but I would guess that it's going to be going through a molecule similar to ATP synthase. They're harnessing that energy, but instead of producing ATP, they're probably producing something else. Oh, the actual hydrogen ions? Yeah, they will be used in the previous parts of the cellular respiration cycle. So all those steps where we're generating NADH, those hydrogen ions will go back to an NADH molecule in one of those previous steps. Yep. Yeah, great question. Any other questions? All right, I like this image. It's a good summary. It shows you where things are located, major products. I really like that. It really emphasizes that the electron transport chain is in this inner mitochondrial membrane here, right? And we have this really big ATP payoff. Now, I don't know if you remember on Friday that I mentioned that other organic molecules can be used in cellular respiration, but often we only talk about glucose, right? So we could use carbohydrates, proteins, different fats, and they can go through the cellular respiration cycle. The difference being is that they're going to enter the cycle at different stages, right? So for example, if we look at these proteins here broken down into amino acids, they will eventually be converted to pyruvate, right? And then they can go through the remaining steps, right? Or they could eventually be converted to acetyl-CoA and then go through those remaining steps. They're just entering that pathway at different points. But we're still utilizing that very same pathway, right? Instead of having a different pathway for carbohydrates, a different pathway for proteins, right? Because utilizing that same pathway really increases efficiency. All right, any questions? I always find it difficult to teach cellular respiration because there's just so much going on. I tried to simplify it. So focus on the major molecules that I highlighted. Focus on the products being formed, right? So how many molecules of CO2, ATP, NADH, FADH2, right? I want you to put this into practice, right? I find value in having you having to actually draw out this cycle, right? So what I want you to do is I want you to draw out this cycle and include all of these terms, okay? I know it's a lot of terms, but I think actually physically going through those steps is really going to help solidify and make things make sense. This assignment I have posted it on Moodle, and there is a Moodle submission site up on Moodle as well. If you choose to hand draw it, you can always take a picture and upload it, or hand it to me in class. Right? But I know digital submission seems to like that in this class, so I added it there. Right? There's a lot going on here, right? But I promise you, this is a great study tool. I do want to reiterate some homework. Right, so content check number 3. We'll be doing that final question in class on Wednesday. Okay. I extended this Nobel assignment because I don't think I reiterated the points and how I'm going to use those points very well. So basically, these are exam points. All right, your next exam will have 43 points on it. 2 of those points to make 45 points will come from that Nobel assignment. Right, so because this is not really an optional assignment, it's not giving you an additional 2 points for your test, but it's part of the test, I wanted just to emphasize that and make sure everybody had a chance to do it. Right? The lectures, they're recorded. You can still watch them. Go to the Nobel Conference site at Miss Davis. We'll have a link to the recorded ones, or you could simply search in YouTube, Nobel Conference 59, and they'll have the speakers that were presented. Yes.
So, for
It's not how it works. Do I need the structures of all the molecules? But do I need like the names in the right places? Yes. So instead of drawing out fructose 1,6-phosphate, you could just simply write fructose 1,6-phosphate. I will post an answer to you after Friday. Yes. And I don't think we'll have time in class to go over it. We will have some time during our exam review day, and I am throwing around the idea of doing an extra review section on a Wednesday. I see nodding heads yes. Would that be helpful for you all? Like if I did an exam review like 4 o'clock on Wednesday or something. Alright. Sounds good. I will plan on scheduling that and letting you all know. On next Wednesday. So the exam is next Friday. This is a very short meeting. That is all I have for you today. You are free to leave, enjoy your extra time. Yeah, it's kind of like proteins that are embedded in the membrane before that it's not electron transport. Yes. 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 ETC is like all those proteins embedded in the membrane besides the ATP. Those proteins that draw those hydrogen ions across the membrane.
As it pushes through, it takes the ATP. Does it take the ion?
It's like the ion flows through the electrons, and that movement is what causes the reaction phase to spin and create ATP from ATP. Yeah. Yes. Everything you're saying is correct. I know. It's a lot. I did put on quite a few like supplemental videos of the work too, if you need the visuals. Yeah.
I'm really gone.
Okay.
Is there anything that I won't be able to about that's that wouldn't be on meeting? It's like I'll watch the lecture and take notes and all that jazz.
Yeah.
I think so. Yeah.
Yeah. And if there is, I will let you know, and you're fine. But I don't think there is. It's Friday. I mean, your drawing is due, corrections on the exam is due, Nobel assignment extension is due. But that's all on Moodle. Yeah. Yep. So, except for the exam. You let me know you're gone. So if you don't turn it in on Wednesday. Any exam corrections?
Oh, I thought you said the exam extension. I was like,
Oh.
you said the exam was
I might have said that. I'm jumbling all my words. It's been a rough Monday. I don't know why.
You're taking years off my life here, girl. Okay, that sounds good.
All right.
Sure, and then I'll like ask the location. Thank you.
For lab today, I'm like unable to use Excel, so I did the best I could. Is it okay if I throw it?
Yes. Can I ask, do you have a Chromebook by chance?
It doesn't have Excel.
No, all right. I've been running into that issue with a lot of my students, so I am working on a way to see if we can use Excel, the online version, to do custom error bars. It's not a simple solution, but it will be a solution.
Google Sheets like has error bars, but then there's like a whole process you have to go through in order to get like individual numbers for it,
Yep.
which is
It is very long. Yeah. Who are you asking? Molecular bio? Who is it? Yeah. I was just curious. Okay. Okay. Let me think about it. I have to go look at how I worded the question again, but I think it might be in your favor. My wording and the evidence you would present to me might be.
I can go find the evidence.
Let me look at it again.
Okay.
Yes. Oh, if you have them, I will take them now. Perfect.
yeah, for this.
Yes.
Possible that I can before
No, so if you look at the email, I sent it out last Friday. There are 4 specific questions that talks about wrong, so that's why I'm giving you an option to review those 4.
Okay, I might.
Yeah, because why not? Yes, on Friday. Let's see. Let me get to my email. Let's see. What's your last name? Oh, there it is. Pop right up. Test corrections. And I will attach the sheet to this and send it out to you right now.
Thank you.
Yeah, not a problem.